Gracias, buenas tardes a todos. Estamos solamente esperando unos par de minutos antes de comenzar la charla del Dr. Chris Beatty. Everyone, welcome. Thank you for attending. We're just waiting a few minutes before we start. So hold on a few minutes and then we can start. As you can see, I don't know whether you can see, but we have some, Chris, we have some people that at least we don't usually see or not very frequently, like Erland Nielsen, Idel Cooper, and some other close people, colleagues. Which is
great!
Yeah.
Right now, I'm just seeing my slide. I'm not seeing the people popping up, but that's fantastic.
Hi, Chris.
Hi. Idel, it's good to hear from you. That's great. Thanks so much for tuning in. It has.
But I'm very excited.
Orlando. Yeah. Well, thanks. Yeah, we should.
I'm not very far from you. I'm in the Bay Area, staying with my.
Are you?
Yeah. So I can almost hear you down the road.
Wow.
But we'll go through Mexico as in.
That's great. Well, after this, let's chat and see if we can set up, a socially distanced get together.
Great, I would love
that. I would love that too. Wonderful.
And thanks for hosting again, Alex, as always.
Nice. No, nice that these things happen. People get together in a way.
Bringing people together.
Very nice. And by the way, Chris, you must not miss Idel's talk, which is going to happen in February.
Oh, wonderful! I definitely will check that out.
Mm-hmm. Mm-hmm. So what happens is usually our speakers and people that attend these meetings, they will receive regularly our announcements of the future seminars. So you will receive this eventually, Chris.
That's great. Thank you. Yes. That's one of the I've mentioned to colleagues with the pandemic coming on, so many things like professional meetings have been disrupted, but a few of the organizations seem to have really handled it well.
Yep.
And I am impressed. For example, the Entomological Society of America is a group that I usually take part in, and they held their annual meeting in November. And their contribution from international attendees just skyrocketed, which of course, doing it virtually really opens the door for that, where, you don't have the finance and the visas and paperwork and all of the other challenges to get to a meeting. So I'm kind of hoping we'll see down the road, once we can all be in the same room together again, that we'll still, maybe do things like have special virtual meetings, or maybe do every other year. Being able to meet in person and chat with someone is very important. I think that's one of the biggest benefits of going to a meeting. But if we can start integrating more virtual meetings and making it more open for people to take part at that level, I think that would be great.
Definitely. I think we should start, Chris. So let me introduce you.
Great, thank you.
So thank you very much again for attending this seminar. We are very happy, extremely honored to have Chris Beatty, partly because he's a great professional, and partly also because he's a close person to me and to many of the attendees, which is really nice. So let me just give you a few words about Chris. He's now working at Cornell University, New York, and he has been working with many issues in odonate and dragonfly biology, ecology, and systematics in general. But he's also a great entomologist. But above all, he's a nice person to talk to interact with. Actually, he, Jason Breed, and Cata Suarez and myself, we are now co-editing the second edition of Odonate Ecology and Evolution book, which should appear at some time in this coming year. So that's why Chris and I have been talking a lot lately, and I asked him to give us a seminar. And so So we can hear you now, Chris. Thank you very much for accepting this, and I'm very happy to have you.
Well, thank you, Alex, for the invitation and the introduction, and thank you all for being here today. This is a topic I'm always happy to chat with people about, and so I think it's an interesting and important one. So thanks for the opportunity to share it. Many of you, if not most of you, let's see. I'm trying to, oh, there we are. Many of you, if not most of you, work on insects in some capacity. And of course, one of the things we know, I very much like this graphic that Kara Gibson put together. I think it drives it home very well. But one of the things that we know is that insects, from a species richness standpoint or a biodiversity or biomass standpoint, are a major part of life on Earth. If we look either at the described species that we currently have, or some of our estimation of how many species are out there, they're a big component of all habitats, and a major part of our living world. We also know, especially in the last few years, that we have reason to be concerned in general about insects, and that there are obviously, you can see articles like this as headlines that are very scary, and there are some people who push back on those as as being maybe a little too alarmist. But I think the data is out there from a number of sources to suggest that that we should be concerned about species distribution, species richness and diversity, and certainly about abundance and biomass, that there are great issues for insects. As Alec mentioned, I work quite a bit on dragonflies, and they're a group that I'm passionate about, as so many people are passionate about their personal group of insects. But I do realize sometimes I catch a little bit of shade from colleagues who work on things like beetles or flies or the Hymenoptera, that odonates are just not, they just don't stand up as far as the global species richness. They're a very old group. We have interesting fossil examples from as far back as about 325 million years ago, precursors of the modern dragonflies. And while estimates vary a little bit, the numbers usually are suggested that there's about 6,000 species of odonates worldwide, and that's roughly evenly divided between the dragonflies and the damselflies. I think there's a little over 3,000 described dragonfly species and a little under 3,000 damselflies. They're a broadly distributed group. They're found, I think, on all land masses except Antarctica. And as with other insects, we're concerned about their persistence and about how they're faring in this changing world. The IUCN has done their best to do some surveying and estimate how things are going. There are about 106 odonate species that are listed as endangered by the IUCN. And their suggestion is that about 10% of all odonate species are at risk of extinction. But of course, that's a challenging thing to say. That could be a gross underestimate because there's a great deal we don't know about current distributions of species. And there's also a lack of information, if you're going to look at change over time, of distributions in the past, so as to be able to do a comparison. One paper that's come out recently that I thought did a very nice job of trying to do comparisons over time was by Joan Ball-Damereau and her colleagues. They looked here in this area where I currently live. They looked at 45 different sites in California and Nevada. And the reason they chose these sites was because they'd been visited by someone named Clarence Kennedy back in 1914 and 1915. And he'd published information on them in I believe, a Smithsonian periodical. And so they had background data on distribution of species at these locations from a century ago or over a century ago. And were then able to go back and resurvey them. And they found a couple of interesting things. One was about 65% of all recorded dragonfly damselfly species had declined as far as sort of where they were found. Though they found that while sort of abundance had gone down, that in some ways there hadn't been such a big change in species richness at a number of sites. But what had happened was basically that some species had been replaced by others, and so they found that another big change was that the overall similarity of sites had increased. So you had these sort of more generalist species that had come in and replaced species that had more specific habitat requirements or specific life history attributes. One thing they found was that species that had that overwintered as an egg rather than as a nymph were more impacted and had declined, perhaps due to their inability to respond to changing temperature regimes. So I was very interested in this, and this has been found in other papers as well, this sort of, accumulation of generalist species and a loss of specialist species, mainly because I do work on these dragonflies that I think can be argued to be specialists in just about any way that you would look at them. And that's the Petaltail Dragonflies. I mentioned that, compared to some other insect groups that, odonate diversity, it's somewhat small compared to things like beetles, but the Petalurids really are a very small group. There's only 11 species known, but for being a relatively small group or a relatively small family, they have an impressive distribution. There are 5 genera altogether. One with 5 species is found in Australia. There are four species distributed along the eastern coast of Australia, and another that is found here on the northwest coast. 2 in New Zealand in genus Europetala. Chile and some parts of Argentina have Phenes raptor, so a single species in that genus. In North America, we have Tachopteryx thorii, found on the east coast of the continent, and west coast of, actually Tanypteryx has 2 species, one found on the west coast of North America and the other in Japan. So a kind of a strange distribution. The 2 that I am most familiar with, that I'll just point out here that I've worked a bit on, one is the Gray Petaltail Tachopteryx thorii. This is data from the Odonata Central. It's a citizen science database where you can log localities. Individuals can share where they're finding things. Blue dots are historical dots, and green are new data. So you can see their distribution. And then the species I've really predominantly been working on is this one, the black petaltail, which extends from Southern California up into British Columbia in Canada. So they have an impressive distribution for some small number of species, and they also have very interesting life history. Most dragonflies complete their larval stage in streams or ponds that can be very specific as to what kind of stream or pond they use. But the petaltails live in these bog habitats, often referred to as a fen. Most of them are found in mountainous areas. And what we have here, there's really no standing water. In fact, they don't seem to like areas where there's a lot of standing water. But the soils are very saturated. There are usually springs that are constantly flowing. And so if you put on your boots and go walking around, you'll make a deep impression as you step, and as you step out, it'll fill with water. 9 of the 11 species in this habitat dig a burrow that they live in. They hatch fairly quickly after the egg is laid, and they maintain a burrow that they live in. So they're sort of the hobbit of the dragonfly world. And some of these burrows, depending on location and species, can be as much as a ½ meter deep. So there's some impressive architecture that they build. The burrows fill with water because the soil around is saturated. And the other really odd thing about this group is how long they live in those burrows. It varies, the estimates vary, but Tanypteryx is thought to live for 5 years between being an egg, hatching as an egg, and emerging as an adult. And some of the Australian species are thought to live even longer. So it's an amazingly long time to spend in the juvenile stage. Most dragonflies complete that within a year or less. So another oddity about this group, you can see an opening. This is just the opening of a burrow zoomed in. And then that's my wife's hand. The muddy finger is the one she just stuck into a burrow to extract the nymphaea. And so we've watched it off. You can get a bit of a look at them. So they have a very unique life history. So we had a few questions as we looked at them. One of them was, how did they get to all these places? One possibility was something like continental drift that, implies something about how old they are, which would be, interesting to find. But the notion that they've gotten there more recently is not out of the question either. Long-range dispersal, as we've seen demonstrated more recently with some migrating dragonflies, is really quite possible. There's a paper here I've cited. This was work done by Anderson and colleagues that looking at Pangaea flavescens, the species called the globe skimmer, which was found back in 2011 or a few years earlier than that, to migrate regularly between India and Africa. They hop, sort of leapfrog their way across the Indian Ocean on islands and rocky outpoints. In fact, that was how this was found. There was a survey going on of, I believe it was on seabirds, and on these rocky points with no fresh water, all of these dragonflies were showing up. It turns out I just yesterday saw an article or a news report about it. I haven't read the article yet. The same group of people have found that the same species apparently migrates between northern India and places like Tibet and Japan. So these are some massively long movements. And so the idea that, over time that, these dragonflies may have gotten here through, through movement is not out of the question. How we chose to address that was to try to look at the relationships between the species. No one had done a molecular phylogeny of the group. There had been some estimates about species relationships based on morphology and also based on behavior. So we decided to go ahead and do this. We were able to get 10 of the 11 species. We had some trouble amplifying one of the New Zealand species. And we have the 8 different replicons there that you can see, nuclear and mitochondrial, that were used to build the tree. Looking at trees on slides are always a little complicated, but if I can, I'll take just a moment to step you through this and what we've found. First, I'll outline the outgroups, all of the things in the tree that we've used for comparison that are in fact not petaltails. We have a few individuals of Tachopteryx thorii, which is the Eastern North American species. We then get another clade that is the Tanypteryx genus, and this one's interesting because we find that the species from Japan, Tanypteryx priori, sort of emerges as a group from within the Western North American Tanypteryx hagen. Then we have another group that has the New Zealand species, but also has some fairly close relations with the species from Chile, Phenes raptor. And then the Australian Petaluridae come out as a group. Now, this might not be that surprising. They're all relatively far from one another. These look to be pretty well established species. Even within Australia, there's appear to be fairly clear evidence that the species are relatively distinct from one another. But looking at the overall shape of the tree, certainly this sort of odd question between what's going on between Chile and New Zealand, and then a couple of other points on the tree that I'll show you. Excuse me. First, there's this spot, which I would call the origin of the Petaluridae. This is the point in the tree that if you go forward, if you turn to the left, you get the outgroups, and if you turn to the right and go forward across the screen, you have just Petaltails. And the other point that becomes, I think, important later is what I will call the Laurasia Gondwana split. So you can see there's sort of 2 major clades, the Tachopteryx and Tanypteryx, and then Europetala, Phenes, and Petaluridae that are relatively separate from one another. So with that data, we decided to try to use fossil calibrations and a program called BEAST to do an estimation of how old these different nodes in the tree were. So this is the same tree. The bars that you see are error bars around those node estimations. And of course, as you go further back in time, those bars can get very large in cases. But you can see the same organisms labeled here, and then our time scale across the bottom. And when we first got these results, I have to admit, I was really surprised. This tree goes a lot further back in time than I'd originally expected it to. And I'm actually going to, if I can, take a moment to sort of walk you through the shape of this tree to kind of better understand what's going on. So first we have our point there about 160 million years ago, where the Petaltails originate. And I'm able to use some of these maps from the Paleo project. This goes back a little further. This is back 195 million years ago. But this would be an awful lot of what, fairly close to what the Earth would look like at that time. The supercontinent Pangaea existed. And so you have Australia, New Zealand actually at that time. The precursor to Zealandia was here near Antarctica and where Australia meets now. And then around to South America, North America, and then what would someday become Japan up here. So that would be what the world looked to. That split that I pointed out, the Laurasian Gondwana split, that comes up. This is a little different time than that, 152 million years ago, but we can see that that split's already started to take place. So we have the Laurasian species here and the Gondwana and sort of along the coast down here. No historical records from Africa and no fossils yet found from Antarctica. But yeah, so that's sort of where our distribution is. As we move forward, we start to see the different genera within the Gondwanan group appearing, and that matches up reasonably well. This is a little further along in time, so the split between South America, Africa, Antarctica, and Australia has really, it's really established. But we see that breakup. And then when we also look at Laurasia, the northern continent, we get some interesting results. First, there's this 103 million years ago, one of the main ones. And that's the split between Tachopteryx and Tanypteryx. And it turns out that in that time period, we had the formation of what is sometimes referred to as the Western Interior Seaway or the Cretaceous Seaway. And so there was a large ocean splitting the area between where Tachopteryx is now found and where Tanypteryx is found. And one of the other unique coincidences is that, as you see labeled here, we have what's the Asian-Alaskan land bridge at that time. And it turns out there's reasonably good evidence from things like distribution of dinosaur fossils that there was a land connection. We know that part of the world has experienced land bridges during more glaciated periods a number of times through history, and it appears there was one at this time. So that matches up nicely with basically the distribution of Tanypteryx priory in Japan. We get a point where our species are diversifying both in North America and both in, maybe I should say, Laurasia and the remnants of Gondwana. We see speciation starting to happen in Australia, and then further break here in North America. And this is that point where we have the receding Cretaceous Seaway, but we're also losing the land bridge between North America and Asia at that point. And then we have one final speciation event about 32 million years ago, a scant 32 million years ago, at where one of the species that lives on the west coast of Australia splits away. So through this period, Australia has been breaking away from Antarctica. I was surprised at these results. I think I would suggest that they match pretty well with the continental drift hypothesis, that if you lay out the map, that probably, quite possibly, based on what we found, that these are very old organisms and that they arrived in their current locations by just writing the continents on which they live. One of the questions we have is, where did the Petaltails originate? So what was the starting point? We know that they, are found in places around here, generally. We used a program called Lagrange to try to make that estimation, and of course, estimating, that far back in time, there's a lot of variability, but the suggestion was that New Zealand was, of the places where they're found, most likely to be the origin point or close to the origin point. again, kind of a strange find until you realize that at that time, remnants of New Zealand were here. And this program also doesn't allow you to look in places or to include places in the analysis where they might have existed, but you don't have evidence for them, where they may have existed and gone extinct, such as Antarctica, which was along that coast and only became the cold place that it is about 34 million years ago, when it completely separated and the circumpolar current isolated it, and it started to become so cold. Interestingly, there was a paper that I found. This was back in 1986 from a paleontological journal. This is a paper describing a trace fossil called a scolithos, basically a fossilized animal burrow. And the authors wrote this paper because scolithos previously had always been found to be associated with marine environments. And they had found some here in the feather conglomerate in Antarctica, in Permian deposits, that were burrows that were in basically a braided stream bed, which would have been up in the mountains. And the authors themselves actually cite the New Zealand species of Europetala and suggest that perhaps this was an origin source for these burrows. They point out that the burrows do look a bit different from modern petaltail burrows, but that's a long time over which evolution can happen. And we know that a couple of the species actually don't burrow currently. So sort of an interesting find. So what we think we can say from the data we have is that petaltails existed as far back as the Jurassic, possibly earlier. Most of the species have Cretaceous origin, and then they likely occurred to their locations through continental drift. This is a strange finding because if you look at the literature generally on extinction, there are estimates of how long different groups will persist. And the estimates vary. For example, mammals are thought to last for about 1 million years if you look at the persistence of a mammal species. Arthropods are thought to persist for about 11 million years. If you take the age of all those nodes that lead to a speciation event and average them, it's 66 million years, give or take, in the tree. So that these species seem to have persisted as independent evolutionary lineages for a very long time. And perhaps that's even more surprising because they live in these really unique habitats. I can say that looking back, for the most part, the areas where they're found have been warm to cool and wet temperate for most of that intervening period. The species found in northeastern Australia, that's not the case. It's subtropical now, but of course, Australia sort of rode its way into the tropics in that way. So it is an interesting, it's sort of a conundrum. Is there something about habitat stability? Is it the minimized competition? Does living in these burrows make them less likely to be eaten? Is it lower mortality? So it's kind of another question to be addressed. I'll go back very quickly to this slide because one of the things I pointed out was that petaltails take a very long time to develop through their larval or nymphal stage. And we have reason to think that the black petaltail, Tanypteryx hageni, that I work on here in California, takes about 5 years. A colleague many years ago collected eggs and actually constructed an artificial fen in his garage and ran water through it and brought them food to eat, other small arthropods. And they took 5 years to emerge as adults, even in that condition. What we're wondering, if you think of things like periodical cicadas that have a long development time, 13 years or 17 years, and then they have an explosive emergence all at once. We don't see that in the petaltails. If you visit these sites each summer, there are adults flying. But if they have, say, a 5-year development time, and that's a relatively fixed time period, you get some interesting things going on where not everyone in a particular vicinity has the equal probability of mating with anyone else. If an egg is laid a particular summer and it consistently takes 5 years to develop to an adult, then that individual's probably only likely to mate with another individual laid as an egg in that same summer. So if it's a 5-year development time, you might actually have sort of 5 cohorts all living in the same place, but separated by time. And we've done some preliminary work to look at that. This is data from 10 sites. This is looking at cytochrome oxidase 1, so a barcode gene, very basic sequence. And each circle you see here on the map is the location of a site. And the number on the circle is the number of individuals analyzed from that site. So you can see those range quite a bit from 5 up to 119 in this very large fen here. And then each of these circles is basically a pie chart, and each different color shows a different haplotype of CO1. A haplotype, what we call in the graph I'm about to show you, haplotype 18 is very ubiquitous, that's the orange. But in total, we had 27 different haplotypes, and 21 of them are present in this one large complex of fens along a creek in Northern California. And not only we have different haplotypes, but we have the map here showing sort of the stepwise changes between the different haplotypes. And as you can see, many of these are very different from one another, in some cases by hundreds of base pair changes. Excuse me. And I realized that when I first made this graph, I realized that the easier way to show which ones were at Cherry Hill in that one location was to show the ones that aren't, which is these. So the ones in a box are found elsewhere. All of the others here, the colors in this case represent a different site, but all of these different haplotypes are found in that single location amongst individuals that grew and developed within half a kilometer of one another at most. We looked very briefly at sort of pairwise FST values between sites. They're quite low. In some cases, we do get some values 0.1, 0.2, but it would suggest that over evolutionary time, even in the complex topography where they're found, that they're still managing to move around. We did some radio tracking with them. I think that the transmitters we had they could fly around with them, but they were a little more subject to predation than we would expect. But from what we saw, it didn't look like they moved from the location where they were captured very much. But again, over thousands of generations, you could get movement up and down streams between fens and move that way. We wanted to try to look at these cohorts, and we had a large collection over 2 summers of nymphs, and decided to try and basically use their size to estimate how old they were. We looked at head width, abdomen width, and total body length. And this is a graph of body length against head width. And what I basically did was sort of look at the distribution. It shows basically a fairly linear growth. Probably some of the noise here is from the fact that these are from different sites which have different latitudes and different elevation. But we get a reasonable sort of upward trend here. And so we try to pick some individuals from sort of the middle of these clouds and compare them, estimating them by their age, sort of 5, 4, 3, 2. And what we found, again, was evidence of quite a bit of gene flow, though still some values that might be a little surprising for what in many cases are supposed to be individuals from the same population. I will point out it's a little odd that there's a 0th here. We had originally 5 cohorts, thinking that they take 5 years to develop. This is because we collected on a second summer, and of course some of the 1-year-olds that we collected that summer had been just laid in the previous summer. So and still there could be some interesting relationships between cohort 0 and 5. So we see a high level of genetic diversity in this relatively isolated group. Indicates there's probably gene flow between sites, and we're not really sure why they have this diversity yet, but might have something to do with their persistence. And one thing I'd like to look at is there any way that these long development times, let's say they generally take 5 years, but there's a little plasticity. Do you have, independent cohorts generating mutations separate from one another that end up being sort of, shared across an evolutionary time? There's a modeling question there to look at, I think. And finally, I started talking about issues of loss and change in the Petaltails. And again, we tend to expect that specialists are not going to survive in the long term. That's things that have a very specific niche when there's large scale change will be lost. That doesn't seem to have happened to the Petaltails yet, but there is, I think, reason to be concerned about our current Petaltail diversity in a variety of places. One is that these habitats in many places, certainly in Oregon or Oregon and California, and in some places like Australia, are subject to the effects of cattle grazing. These are very green sites because there's always water. They have a lot of specialty plants. It turns out there's a lot of carnivorous plants that are found in these fens, but there's abundance of plants there, so potentially useful for grazing, and also as a source. The springs tend to be permanent springs that have a lot of flow. And so there's concern that these are, the water's being diverted, or cattle are going in and, stomping up this soft substrate and, potentially damaging burrows and killing individuals. Logging is also an issue. A lot of these populations are in national forests, which are subject to logging. Different times, different presidential administrations, I might say, are more or less inclined to promote logging. So we're in a period of large-scale logging here in the U.S. And this again can really change, especially down gradient hydrology can change what's going on in a stream. And of course, major issues that we've been having, certainly here in California, are both wildfires and drought, which can definitely be associated with one another and with climate change. But as you see, these are some estimates from just a couple of months ago, sort of late summer. We have an extreme, this is prime Petaltail habitat here in California, the extreme drought showing up, and then the distribution of our fires. This is the Sacramento Valley, or the Central Valley, here in the middle of California, and a lot of areas, again, where fire threat is great. Whether or not these habitats being so wet would make them resistant to fire damage, I don't think that's been looked at. But of course, it's not only a concern here. In the last year, Australia has experienced some terrible wildfires as well. And again, when we look at their distribution, they're showing up. This is New South Wales here, where Petaluridae gigantea, a species that is listed as at risk in that Australian state, is found. So it really comes down to some questions about habitat management. A lot of these sites aren't well known, certainly in things like the national forests and national parks. And fortunately, there's been a big effort in the last decade to identify those and to find them and sort of manage them. These little fens can sometimes be quite isolated, but there have been survey efforts that have been trying to identify these, what they call water or groundwater driven environments. And this is, previously, there had not been a lot of effort to look. There had been some surveys for specialist plants, but not for these dragonflies. And so I've been working with some colleagues at the Forest Service, trying to do that and get a better handle on where they are. A lot of it comes down to the maintenance of the hydrology. As you can imagine, if these things live for 5 years, if it dries up one summer, then everything dies. And so keeping the permanent water flowing naturally is a big part of keeping these habitats safe. And then there's the question, if we're going to manage, from a population genetic standpoint, we're going to try to understand a little more about what's going on with this population structure and kind of get at what is a population in these species. And that's what we're doing now. We're moving forward with doing some SNPs and potentially some whole genome sampling, starting work on a de novo genome assembly for one of the Petaltail species. And as I mentioned, surveying, trying to find locations and really get an idea of where they are on the landscape, and also potentially do some water chemistry work, looking at what's unique about these fens as far as the the habitats and then look at potentially eventually look at genetics and larval development. Is there, why do they take 5 years? Is it some sort of, environmental limitation? Is it genetic? Possibly a combination, but we'd like to tease that out. And I'll leave you with just one unique moment that when anytime I stare at this tree, gets me thinking about it. There's that spot right there at about 100 million years ago, where the gray petaltail here is separated off from the western petaltails, and say that would suggest that this species has been around for 100 million years, and there aren't too many. I mean, we have coelacanths that we know of today, but the extant coelacanths, we don't think those species have persisted that long. It's just the group has lasted since the Devonian. Maybe horseshoe crabs, but as far as a species that's hung around, I don't know of many examples that would rival this. Just sort of an interesting thing to ponder. And with that, I've had the pleasure of working with many colleagues, friends, students, spouse, and I hope someday soon to get my son out helping with these. In fact, he did come out and help do some sampling a couple of months ago. And with just a lot of colleagues and institutions I'd like to thank for support. And I'd like to thank you for taking the time, and I'll be happy to answer any questions.
Oh, thank you very much, Chris. Yeah, we open this for questions. You can either use the chat option in this Meet program, or you can directly talk to Chris and ask for the microphone in that case.
Sure, I'll pull this up. See if I can put this. You are still seeing the screen, is that right?
Yep.
Sounds good. Okay. And then.
Hi, I have a question. Hey, this was awesome. I'm from Bolivia, and here we don't have so many works like this. Well, my question was, well, can you explain again what is a population in this species?
Well, that is a good question, and it's our evidence suggests that there's some movement or some gene flow between, and if it wouldn't take. So let's say that for some reason, development time in these species is absolutely fixed, and every individual takes 5 years to develop. That that could lead to a situation where you get no gene flow between cohorts, and so you would have sort of 5 parallel cohorts in any one locality. And in that case, I think you would have to think of those as 5 separate populations. And whether or not we should think of those as 5 separate species, then that would take a great deal more analysis. But yeah, that's sort of one extreme, where you might think that there are multiple populations basically living alongside one another that are just separated by time. But we, if you had some gene flow, it wouldn't take a great deal of gene flow, a little bit of plasticity. Some individuals that took 4 years or took 6 before you had gene flow, and they still sort of maintained as one big population, although potentially with some unexpected structure.
Thank you very much. So Yesenia Vega has just written a question. She says, have you checked for contaminants or NUMPs in your COI data? Because the difference between haplotypes is huge. I haven't seen this in other odonates.
That's a really good question, Yesenia. And that's something I say with this first, this data was very much a first pass. And yeah, it is very odd for other odonates. And an example would be the work that Manfred Kohli has done with some of the Somatochlora salbergi and others, where you have these circumpolar dragonfly species that are broadly distributed, and there's virtually no diversity in looking at something like this. And of course, the real problem is that we really shouldn't rely on CO1 data at this point. It's a fairly short sequence, and so that's why we want to. This is a surprising result that definitely needs to be examined further. We have this data. I'm not the graphs that I showed you, like the haplotype maps, aren't published. That's just something that we have, and I think was useful to look at. We also had, I didn't show it, but we also did histone 3. So we have CO1 and H3, and the H3 show a very similar pattern of surprising diversity. One of the things we may need to get at as we get into, we right now have a rough estimate of the genome size for Tanypteryx hageni. We're right in the middle of working on that, but the first estimate we had back was that it was about 2 gigabases, which is, for what we know of estimates for odonates, very large. There's not a lot of data out there, but most of the estimates are like 0.8 gigabases or 1. I think some of them, like for some of the echinids, it's up to like a 1.2 or something like that. We're going to continue that and use some different calibrations and see if that persists. But one question we have is, is there something like a great deal of gene duplication? So numps could be a real concern, and that's something we'll want to, as I say, as we get into trying to do things like whole genome sequencing or doing RADseq, that hopefully we'll be able to better address. But it's a very important question, and it's not out of the question that that's what's driving it. Though I'd be very interested to know that as well, and see if if these things do turn out to have these inordinately large genomes for dragonflies, see if it is that, see if there's just a lot of material that's built up, if they're just why they've expanded so much.
Thank you, Chris. I have a couple of questions. So it is my impression that this is that cattle grazing would be like the first time that I see that as a source of stress for odonate larvae. I try to recall that even from Corbett's book, trying to see whether he even mentioned that. But it seems to me that there are not so many pieces of information in regards to that. Or do you have any evidence of any other odonate? I mean, it seems like, especially given the place that they use as a habitat, it overlaps so much with cattle's, that explains that, because it doesn't seem to be the case for me. I mean, I'm sure that there may be other species in other places in the world, because cattles are everywhere, literally. But it doesn't strike me as nothing published in relation to that.
Yeah, and that's I think would be very unique to these dragonflies, though not exclusive. And that's one of the other things that down the road we'd like to get into, because there are examples of other fen-dwelling species. Somatochlora has some. That's probably the best example, certainly of a sort of a North American example, is Somatochlora Heinei, I think, Heinz emerald, which is found in the Midwest in the U.S. That's the only, I believe, the only federally listed endangered dragonfly in the U.S. And the reason being is that they live in fens or live in bogs. They're found in from sort of I know of locations for them in Missouri, sort of moving north, like up through Illinois into Indiana, and there's a couple of examples specifically in Illinois that I'm familiar with where I think it's a place called Ford County. There's one particularly very strange sort of elongate county in that part of Illinois. There's a lot of farming. It's, you know, a huge, it was old tall grass prairie, a lot of farming in that area. And the reason Ford County was the weird shape it is because it was all sort of a big fen area and no county wanted it. And so it ended up, finally, there was a mass effort to go in and drain it and then it became very rich farmland. But of course, then it really hit the Heinz Emerald. They also have a larval stage that lives in burrows, though they have a weird relationship with crayfish. So they, I don't think, dig their own burrow. And they are also known to take, I think, 2 to 3 years to go from egg to adult. And we have some other, so Somatochlora has some interesting examples. And another genus, the Corduligaster, the spike tails, has some interesting examples. They're variable between species. Some live in streams and some use more pond habitats, but there are a few that use these fens. John Abbott, who's now in Alabama, was originally doing work in Texas, has described species called Sarracenia, which is found in these fens. In fact, he finds Tachopteryx nymphs right in with them. And it was a unique story because they had originally been lumped in with another species, but turned out that they had this very different larval habitat that they used. And so, so that, you know, there's some interesting comparative processes there that we could look at, especially in these, you know, we have the Petaltails, which sort of all have this unique habitat that they use. Though I say some of them don't burrow. We're suspecting that that's a loss over evolutionary time. The two that don't burrow, one is in the Laurasian clade and the other is in the Gondwanan clade. Although they live in fens, and they create sort of a little pocket. There's a paper by Ian Baird that's come up fairly recently. I think that was in International Journal of Ornithology, but I have to check that. But Ian has sort of documented some Tachopteryx habitat and really looked at where the nymphs were spending their time. So they don't, you know, they don't dig a vertical burrow like the others do, but they still have these little areas that they spend their time sort of under the detritus. So yeah, I think there's some interesting comparative work to be done there, especially in these groups where some live in bogs and some live in other habitats.
Yeah. Thank you. No, I was thinking about like trying to link these different life history traits and the risk, how threatened different ordinate species are in terms of these life history traits. Like, for example, what you were saying, how long they spend as larvae, and the type of microhabitat that they use. I mean, you know, from Viola's famous paper now, like 10 years ago or something, about how threatened odonates are, I think it would be nice to see like a sort of a reassessment of these different life history traits, how much they can explain the likelihood of these animals, of the different species to be threatened. That's one of the things that I was thinking when I heard about this impact by cattle, and so yeah. So thank you. So Idale Cooper, she has a comment. She says, I'm wondering if fen-dwelling species have longer larval stages than average. I think a comparative study would be great, like you just described, Alex.
I agree completely. And yes, I think the evidence that we have so far suggests that might be the case. I mentioned Somatochlora, the Heinz Emerald. I think it is known to take 2 or 3 years. I don't know if there's a lot on the Sarracenia, that the Corduligaster Sarracenia that John described, but John and Kendra Abbott and some colleagues have been further working on that. I think that there's a possibility, and then comes the question of, does that have something to do with the habitat? For example, as I mentioned, that the fens here have a lot of things like carnivorous plants, very specialty plants that are often found in habitats that have some nutrient limitations. So is there something, is there a trade-off? Is there some strange thing, some reason why it's good for these dragonflies to be in fens, but that their development time is impacted because of some environmental limitation? And I say this is weird, as you mentioned, Alex. You don't really have to think usually of well, maybe water extraction for other species is a concern, but that cattle aren't as much of a problem, but they can really devastate one of these. Group of cattle can go in. I've seen some sites where there is a sort of an identified spring where there's a place where the water's coming out of the ground, and there's a containment built around that, like a cage, to keep the cows out of that. But then they're sort of stomping all around it, and it takes a very small amount of time to really go in and just tear that habitat up. But yeah, I think the suggestion already is that there is something weird going on between fens and bogs and obligate dragonflies that use them in their development time.
Yeah.
Oh, Adele, yeah, you mentioned interesting in damselflies too. That's true. Yeah. And we do find there's some Argia that there are some damselflies that we find in and around these fens. I don't know if I would be inclined to describe them as obligate in using them, but that there are other species. We see a lot of other dragonflies flying, but of course they're using these open spaces just as adults as sites to forage. Oh, are those the Megalagrian? Do those have some bogs?
Yeah,
some of the Hawaiian ones. We should definitely talk about that. And of course, Megalagrian is interesting because there's a lot of examples of ecological diversification there where they've specialized.
Yeah,
so
habitat variation. But like you guys were describing, I think it would be really interesting to see whether the species, like what are the correlates between the species that are maybe threatened with climate change more, if they are more specialized in terms of larval stage length.
Yes. Yeah. And that's a lot of that is data we could probably go ahead and get. Obviously, there's some issues like with the Petaltails. There have been some suggestions that some of the Australian Petaltails take 20 years. But that comes with there's the suggestion's also been made that might be flexible, and the reason it's flexible could be as a drought tolerance. That when you have intermittent drought, that basically what you have happen is on years that are sufficiently wet, that individuals will wait and emerge at those times. Gunter Theisinger, who works there, has mentioned that there are some of the species for which he finds adults that are very different, especially the males, that are very different in size. So you get some really large adults and then some that are like, I think, as small as like half the size. And so, that might be, I think his sense is that that's not a different species, that they're still part of the same species. They're found in the same place. So that could be a weird case where you get, if it's a drought tolerance, where you get an individual that spent 8 years as a nymph and another one that spent 17 years as a nymph. Yeah.
That's so fascinating that they're that long as larvae. And it's really interesting to think about how the longer they spend as larvae, the more likely it is something will happen before they emerge. But also, the longer they spend, the more flexible they are to wait for prime conditions before they emerge.
Potentially, yeah. So we might be seeing the habitats I've been looking at, historically did not deal with quite so much in the way of effects of drought. They're probably going to be more impacted, sadly. Well, they're probably more impacted now than they were 20 years ago and will continue to be. But yeah, so that's, being able to do that and sort of time my good colleague Steve Valley, who was the person who really sort of introduced me to Petaltails when I was a master's student, made an observation at a Petaltail site up in Oregon where he'd gone to visit it one summer, and they'd logged just above it, and everything had dried out. And he thought, well, that's it. This site is gone. And he came back the next year and found adults flying, but 5 years later he didn't. And so he had speculated that it could be possible that one cohort was badly impacted by the extreme shift in hydrology that particular year, but maybe there was still enough water that, that the others down in their burrows had survived. Totally anecdotal, but kind of an interesting observation, something, again, that suggests that we definitely need to dig more into their population genetics.
That is so cool. Thank you, Chris. That's a great talk.
Sure.
Yeah, I agree. Yeah, that was a great talk, and I'm pretty sure that you enlighten some people from different places, and I'm sure they're gonna go out and look after these species to see whether they are similarly impacted or not, depending on the habitat that they live. So if I wonder if there is any other questions, comments that our public has.
Everyone should feel free to email me as well. Just if questions come up, please reach out.
Can you actually write down your email address on the chat,
sure,
please?
It is. Well, I can give you two. So I have. And also.
Right.
When I was looking for a Gmail account, I decided that the genus name was probably not taken. So, yeah, I ended up with tanypteryx@gmail.com. But feel free to reach out with either of those.
Okay, then. Thank you very much, Chris, again for talking today. It was really nice, and it was nice to see so many people that I didn't know, actually. I mean, people and from different places, people that I think attended this for the first time, which is really nice. And so we'll be in touch. This is our last seminar in the year, so we're gonna start again in the following year in February. Actually, Idell Cooper's talk will be the first one. That will be in February. We will let you know the exact date in due time. And so thanks, Chris. Thanks, everyone, and have the best end of the year ever, and despite the pandemic.
Yes, thank you. Thank you all for the opportunity to do this and for the great questions. And yes, please, whatever holiday it is you celebrate, please make the most of it. And a good new year to you all.
Thank you very much. See you, everyone. Bye.
Ciao.